So Canada geese, we are all familiar, I'm sure, with them. We've all probably seen that distinctive flying V. You can hear their honk from a mile away, and they are a very large bird, so they're hard to miss. So Canada geese have a lot of different subspecies of Branta canadensis. So there will be a variation in size. So some can be a little bit smaller, and they can get to be quite large within these different subspecies, as well as variation in color. But you will see they're all still going to look, for the most part, you'll be able to identify Canada geese from one subspecies to another. They all have that long dark neck, that white chin strap, and of course that grayish tannish body. Geese, in terms of habitat, like the land-water interface. So they need both land and water for their life cycle, and we will find them, of course, within that area, regardless, as long as there's water, food, access, safety, all of that is where they will be found. In terms of food, they are a relatively opportunistic feeder. So they eat grasses, sedges, berries, grains. So a lot of different foods. In an urban setting, we've probably encountered people who are feeding the birds their bread or their hot dogs. Geese will likely go after those too. It is highly discouraged, so do not do that. But again, they will likely go after food of that as well. And then in terms of nesting, they do mate for life. Canada geese do, reaching sexual maturity at about 4 years of age. Their clutch size is about 2 to 8 eggs per nest, and those young will stay with the parents for about a year before they fly off and find their own family. So they, that is quite a big group of these large birds that we can find out just in a given family. So why are geese important? So Canada geese are native to North America. They went nearly extinct in the early 1900s. Does anyone want to guess why?
I have no idea. Back there was nobody around.
Well, in the early 1900s, it was over harvest, so hunting.
They hunted them.
They hunted them almost to extinction, as well as wetland loss. So that plummeted their population, and through recovery, through conservation efforts through the DNR.
Yeah, but they breed like crazy.
Yes. We have been able to bring those numbers back. So they are a conservation success story in a lot of ways, and now we're just trying to work on kind of manage those populations and interactions between geese and humans. They're of course vital for wetland ecosystems, for seed dispersal, nutrient cycling are important for those purposes. And of course hunting. Hunting is important for our economy. People hunt for subsistence, just to be able to live off of. I will say my family is a big duck and goose hunting family. And if I had goose jerky, I was going to bring it for them to try, but we're fresh out. So
I've never had one.
So maybe next time. So important for our ecosystem. So when I talked about how those numbers were able to bring back, or be able to jump back so significantly, and I think one of the statistics I read said that from 1966 to 2019, the goose population rose 7% annually every year in that time. So that's quite significant. And I believe the estimate in Michigan is that we're over 300,000 geese. DNR would prefer them to be more in the 175 to 225 range. So they have really exploded in a lot of different ways. And so aside from just us being able to establish hunting seasons and regulations to manage the population, or to provide them to come back and then also manage them, and also with wetland restoration and protection efforts, we have kind of helped rebound that population. But in addition to that, climate change we have to thank for that, which I say sarcastically. Because milder winters have allowed the geese more access to habitat. So geese do migrate, but there are also geese that do not migrate. There are some that do overwinter in the area that they live all year round. And that's because winters are just not as harsh as they used to be. So with less snow on the ground, they're more readily able to access their food all year round. So they like a lot of grain off agricultural fields, like that waste grain. So what doesn't get collected in that agricultural process, if it's not covered up by snow, they can still eat it. So why fly south? So climate change has really done a lot to help reestablish this goose population, and maybe more so than we would really like. But then also as a society, we really love those green, lush, open spaces. So think golf courses, parks, waterfront properties. We have provided, we as humans like it, but geese also love it, and it's exactly what they're looking for. It's that open area where they can see a long distance, they know that there's no predators coming after them, it's safe for them. And so we, given that we have, covered everything up with just some turf grass, they love that, and so that's also really helped and benefited bringing geese populations back as well. Of course, that brings then the human-goose conflict with it. I know that we're going to hear more from the health department as it relates to the poop in molting. So aesthetically, we don't want to go out to a park and find goose poop everywhere. A goose can produce 2 pounds of feces per day.
A day?
A day.
I know, they
yes,
just
it's quite large, and so no one wants to be able to go out and have to walk around that, especially because they're traveling in groups of this size. It's usually not just one goose, it's usually a whole family. And so that can be pretty significant just in terms of being able to enjoy, these resources, these parks and stuff. But also when that excess feces washes into a water, it can lead to things like harmful algal blooms, it can release bacteria, toxins, things of that nature. So then there's a human health component to that, which is why we invited the health department here today.
That swimmer's itch come from that too?
Swimmer's itch does not come from the geese.
Okay.
It does come from the other waterfowl though, in combination with snails. So there's something to that. Of course, crop damage. So when you get a large flock like this, they can do some significant crop damage, but also they're just aggressive. Anyone who's encountered a goose knows that they will chase after you, they will bite, they will honk, they will hiss, they will do all the things. So they are not, they're not safe for people to really be around or try to interact with, as much as people really try to do that sometimes. But when we have these urban environments where we're basically setting up exactly, the prime habitat for them, but also a place that we want to enjoy, we're going to encounter those issues. So the crux of why we're here today is really to talk about natural shorelines. So by installing a natural shoreline, which Noah is going to talk about later, we are breaking up that visual barrier for the geese. So if they are not able to see that long distance, if we have planted grasses and shrubs that impedes their line of sight, then they no longer feel safe, and they're not going to be accessing those sites. And not only that, but natural shorelines also protect your property from erosion, so it is a win-win on multiple ways. Of course, population management is something that the DNR deals with in terms of hunting regulations. And then, of course, there are deterrents. I am not that familiar with all of what is available in terms of a goose deterrent, but sometimes you'll see out at locations that have geese problems, they'll put like a predatory bird statue out there. Like if you go the site we're going to today, they have an owl sitting out on a post. They've had an eagle that flies around. I've never seen that work.
Never. I tried all that stuff. The geese will poop on them.
Yeah, yeah, exactly. They walk right up to it like it's nothing. I've seen the coyote cutouts. So maybe someone's had success. I just personally have not seen that. So in combination though with a natural shoreline, that could be something that works. So without further ado, I am, as I mentioned, if you have questions, come ask me later today. I am going to introduce the health department. We have Jeremy Fruch and Amanda Potter to talk about the health department's beach monitoring
Are you going to talk
program.
more about natural shorelines later?
No, and then Noah will be talking after the health department, yes.
Okay.
So we'll be hearing more about that.
I don't know if you want the clicker, but I do have the clicker.
Okay. All right, so my name's Jeremy Fruch. I'm with the Health Department Northwest Michigan. I'm the Environmental Health Director. What I do is the Health Department, we monitor the beaches, and then we provide these advisories to the public when we have elevated E. coli levels. This is Amanda Potter. She's the lab director or the lab supervisor for the Health Department Northwest. She does a lot of the testing, the actual testing for E. coli, and they also work with, we have a couple techs that do MST, which is source tracking, and that actually will provide what species it is actually we have targets, but I'll get into that a little later, so. All right, so what is the beach monitoring program? So 49 beaches that we do. We do 4 counties: Antrim, Otsego, Emmett, Charlevoix. And what's important here is when it says E. coli levels, when we're testing for these, it's not necessarily the E. coli that is the issue. I mean, it is bad, but this basically kind of like it's a target that says, okay, we're above 300 on E. coli. There's also a risk for other illnesses too, gastrointestinal illness, viruses, and what we do is we do this for 11 weeks. So we're going from June, middle of June till Labor Day this weekend. This is our last weekend. I know it doesn't feel like summer, but
Yeah.
but it's definitely
It's up north.
yeah. So we try to wait till as late as we can in June because we know people aren't swimming early in June. Some health departments go a little earlier downstate, but we wait because we know the weather is hopefully going to get warmer after. So yeah, so this is our last week this week, so it's kind of bittersweet. So again, these are the counties we do. I have 2 beach techs. So what they do is one will start in Emmett and they kind of follow a route like here. They'll do some of the, well, they actually will go up here, up to Mackinac. They'll do beaches up here, go over to Sturgeon Bay, kind of drop down, I think Thumb Lake they do, and then they kind of do some of the inland lakes in Otsego. Another sanitarium will start in Charlevoix and kind of drop down through Lake Charlevoix, do a couple Lake Michigan beaches, go down to Antrim, do some Torch Lake beaches, and then kind of swing around through Bel Air and then get to the lab that way. All the tests or all the samples are collected. They're usually in the lab by 12 o'clock. We try to run these as soon as possible so we get results by 6:30 in the morning.
They go to Beaver Island?
No, we don't go to Beaver Island. There's funding limitations on that. They have tested Beaver Island. That's when they had somebody, I think, living on the island, and they would just kind of ship them off one day a week. But right now, we don't have that luxury, so we're kind of leaving Beaver Island on its own. So what causes the elevated bacteria levels? We actually up here, we're kind of lucky. Our beaches are very clean for the most part. I mean, downstate, I used to work at Oakland County. I mean, the beaches, a lot of times, there was constantly high E. coli levels in a lot of the beaches down there. And what is the 2 main sources here? What we've been seeing is just based on our MST results, we can say that wildlife, waterfowl are the main contributors. We haven't had any human targets actually hit in the last 2 years. So we kind of, by looking at saying, okay, we don't have humans and we don't have bovine, we know that it could either be waterfowl, wildlife, and what's the other one? Gulls. So should not say Canadian geese, because that is a pet peeve of mine. Canada geese they're probably our main culprit in the high levels of E. coli. Our beach techs, they go out, they see the geese in the area, and they're usually they'll report on it, yeah, there's geese out here. And what happens is they go in the water, but they're also on the beach, and they're just going around back and forth, just going poop most of the time. And when we have heavier rain events, we've noticed that the day after, we actually will collect samples, and we'll have spikes in E. coli after heavy rain events. And that's usually because everything gets washed off the beach, in that beach area, especially cold beaches. The water can be stagnant, and the only way to actually get rid of the E. coli is you can kind of do some prevention as far as combing the beaches, but there is, I'm not going to get into it, but I know the next talk, I think it is, they're going to talk about greenbelt vegetation, And that does help manage some of the geese issues. All right, and I'm gonna let Amanda talk about the lab part of this. You want the clicker?
Yeah, that would be great, thanks. As you said, I'm Amanda Potter, I'm the lab supervisor. A very involved lab supervisor, we wear multiple hats here. How are beach samples analyzed? So the main way that we analyze these beach samples, they come into the lab and we add basically food for bacteria in there. It also has a fluorescent compound in there that only E. coli can process. We use Cola Alert 18, that's just the food. The nutrient packet adds the fluorescent probe, and then the sample's poured into a quantitray. So we mix it in the bottle, and then that bottle is then poured into this quantitray, and that quantitray is important. You'll see in the next slide, it helps us quantify how much bacteria is really in that sample. We use a UV light for the glowing wells. It also does coliform, which is yellow. We're really looking for E. coli at this point for the beaches. Most water has coliform in it. The statistical method to determine the amount of E. coli in a sample is called the most probable number. It's just a fancy statistical calculation that was figured out with a lot of test tubes and a lot of different concentrations of bacteria to really enumerate how much bacteria is in these trays. So we count the wells, and then the number of small wells and the number of big wells together give us an estimate of how much bacteria is in that sample. So for 2024, I put the results up here. Richard I. Park, which is in Bel Air, was our biggest beach that closed the most. So the beach that closed the most frequently at almost 20 closures. We had a total of 28 beach advisories, including 2 closures. 6% of beach samples surpassed the limit of advisories or closures. It's a very small percentage. And then Richard I. Park, as I said, was the #1 culprit. August by far is our the month that seems to have the most positives. It's the end of the year, the water's getting warmer, lots of people have been on the beaches, geese have been there a while. But I will tell you that August in this case has really been driven by that one park, Richard I. For 2025, our beach advisories, again, Richard I. Park was our highest. These numbers are a lot lower, as you'll see. There was only 2 closures at this point. This was only through July. I think as of now, we've closed Richard I. Park probably 5 or 6 more times. So add that to that number. The total of 7 beach advisories, including 3 closures. We have Wooden Shoe Park, which I believe is in Ellsworth, Zorn Park up in Harbor Springs, Odin, East Jordan Tourist Park, and then Elm Point Beach. Where's Elm Point Beach? Is that in
Charlotte.
That's Charlotte,
Yeah,
okay.
that's off 66.
Oh, okay, well, there we go. Elm Point Beach. So only 3% of these samples reach the threshold for advisories or closures as of July. I'm guessing that's going to be closer to the 6% as well with that Richard I. numbers this past month. Yep, so you can see an increase to June to July, and then August would be a lot higher again. So another way that we analyze E. coli, so we have the NPN, or the most probable number that we use those quantitrays for. In addition, we do PCR. Polymerase chain reaction is just a fancy way of saying we take a piece of DNA and we multiply it lots of times so that we can see it. It can be correlated with the number of bacteria, and the idea is eventually to go to qPCR to predict outbreaks in E. coli at a lot of these beaches. We're still in the research and development phase, so we're still working on things, but it's also known as real-time PCR. It gives us a lot faster results than a lot of the other things that we've done. And this also gets correlated to the NPN, just like the quantitrays do. This is just an example of an amplification plot for those of you that like to get into the science stuff. These are negative controls over here, and this is where you find a positive over here. The cycles are just the number of times that they're basically, so it goes through a heat cycle to amplify the bacteria, basically to grow it. And this is how many times they have to grow it in order to see the bacteria. So these have more bacteria in them than ones that would be in the 30 range. Does that make sense? Okay. So Coal Alert is our original way. qPCR is what we're trying to develop. Some of the advantages of Coal Alert 18 is it's well established. It takes 5 to 10 minutes for prep. It's 18 hours for results, which is really a long time. It's a downside to Coal Alert. It is less costly, and next day results can lead to harmful exposure. So a lot of those beaches are sitting for almost a day before we get any of those closure signs out. qPCR is about a 2-hour prep time, and then 2 additional hours for results. So we can literally get results in the same day with qPCR versus the Coal Alerts. It is more expensive right now because it's still in research and development. A lot of these things there's just not as many places to get materials and things like that. And samples have to be there a little earlier in the day in order to have time to process by the end of our workday. The goal again is to move away from the Coal Alert and eventually get to the qPCR. Now, the most interesting part of all this is the microbial source tracking, which Jeremy alluded to in his previous presentation. We use microbial source tracking to basically identify where the E. coli is coming from. So anytime we have a beach that's over 300, which 300 is our threshold of putting an advisory up, whether it's minimal body contact, no body contact at all, depends on what kind of advisory, but 300 is our limit. This is digital droplet PCR, which is a little different than the other qPCR than what we did. It's more accurate, and you can see the results a lot easier, which you'll see in just a minute. We use that to identify different fecal sources of water contamination, and it uses microbes that are typically found in the guts of different animals. So if you take a goose, for instance, it's going to have a different percentage of microbes than what a human would have. So it might have certain strains of E. coli that are a lot higher than humans would have. And so based on that data and on those ratios, they can really use that to identify which microbiomes which animals these microbiomes are coming from.
The DNA of feces.
Basically, yeah. So your feces, so like everybody's guts have lots of different microbiome, like they have lots of different bacteria in them. And depending on what you eat, it depends on your species. Mostly it's driven by diet. But so we can use that, yes, exactly. It's DNA of feces. Basically how digital PCR works, so digital PCR is basically it's an emulsification, and what that means is there's lots of little oil droplets in water, and it takes a little bit at a time and basically runs that PCR experiment we saw in the previous slides. That is why it's more accurate, because it's able to break that sample up into lots of little pieces versus just one sample to run. It runs, I believe, 20,000 reactions out of each well, which is a lot. So targets, as far as what we do in the lab, we have to choose which targets we want to identify. So we have to play a guessing game, an educated guessing game about what kind of things we think might be driving the E. coli. So we typically test for cow and human markers. It's typically, if it's not one of those, we assume waterfowl. Waterfowl, though, our marker for waterfowl currently is just that, it's waterfowl. It doesn't tell us specifically that it's geese or gulls or whatever. But we can identify based on what animals are near the beach, whether they're farms or wastewater treatment plants, over what we're going to run and how we're going to compare it with which targets. This is the ddPCR results. These are a negative. This cloud down here is negative. And then this is kind of our threshold line, so anything above that is going to be positive. This is a sample. We might get a few. The positive control has a lot up here, and then this is just a negative control. You'll notice there's no blue dots up there. It's a very yes or no answer. And I think that's it. Are there any questions? No, there's a lot. Yeah.
So if you guys run the so if your E. coli levels come back above 300, it triggers you to run the MST. Is that correct?
Yes, we don't
Okay.
run the MST though. I didn't say this. We don't typically, just because of funding and staff limitations, we save all the samples that have 300 MPN and higher, and then we run the MST after we close the beach program. So
Okay.
we'll start doing that in the next few weeks.
Okay. And then what do you do? What do you then do with that data? Like, how is that used Like,
We,
if you-
so if we find any human markers, then we try to, like, for instance, like if there's a bathhouse on the beach, we want to make sure the bathhouse isn't leaking. So we may dye test the bathhouse if we have any human markers and things like that.
Okay.
See, one thing about the waterfowl marker, it's sensitive, and we've heard that it doesn't work as well. So we are like, let's not waste our money on that, because we want to make sure humans are not the main culprit of
Right.
it. The one target I wouldn't mind looking at maybe next year or the following year is canine, because canine can also be a source. I know I went to a conference last year, and Mark Hansel from Oakland County, he actually found a lot of canine
People don't pick up their
markers.
poop on the beach.
Exactly.
Yeah.
You know, so that would be a good one to kind of just put in there too, because that's a very good marker. They say that works well. Not every marker works the best, so that's why we just went with human and cow, just to make sure agriculture wasn't a prime example.
I should have been clear that MST is
No,
also
that-
in
that-
research and development.
Yeah,
They're
yeah.
still working on it.
Yeah, so,
But,
But people should pick up after their dogs on the beach.
yeah.
yes.
course.
I did
Yeah,
have a
but
couple
the-
more slides if you wanted me to.
Yeah.
Could that, excuse me, could that also be a septic tank issue?
It could be.
Okay.
It could be, yeah.
Just